Hello, everyone. Good afternoon, and thank you for joining PPMD this afternoon for our webinar with Avidity Biosciences. Avidity is joining us today to talk through EXPLORE44, their clinical trial that is an ongoing phase 1/2 trial evaluating AOC 1044 for individuals with Duchenne who are amenable to exon 44 skipping. So we're joined today by Kelly Dystrophin, the Vice President of Medical Affairs at Avidity, and Mark Stahl, the Senior Director of Clinical Development. They're going to talk through a little bit of the science of AOC 1044 and talk through that clinical trial. At the end of the presentation, we are going to have time for some Q&A. So if you have questions while the presentation is ongoing, please feel free to put them into the chat box at the bottom. We'll try to get to as many of the questions as we can during the Q&A. Also, just as a reminder, we are going to be recording this, so if you have any friends or family that this would be relevant to, please let them know. It should be up on PPMD's website within the next week. And with that, I think we can get started for today. So I think, Kelly, I'm going to hand it over to you.
Awesome. Thank you so much, Eric. I'd like to start today by thanking PPMD for inviting Avidity to join you and share an update on our progress on developing potential treatments for the Duchenne community. We recently announced the initiation of a clinical trial called EXPLORE44. You see the logo on the slide here, investigating the potential for our treatment, our investigational therapy, AOC 1044, to potentially treat individuals with Duchenne mutations amenable to exon 44 skipping. Can you move to the next slide? Quickly, I will be making forward-looking statements, as will my colleague, during today's presentation, which essentially just means that we are not making you any promises that this will actually show great results. We are just here giving you an update as to where we are today. On the next slide, I've just got an overview of our agenda. I'm going to be giving an overview of who we are at Avidity, our technology, and our approach to patient-centric drug development. And then my colleague, who you heard about already, Dr. Mark Stahl, will discuss the specifics about our AOC 1044 program and the EXPLORE44 clinical trial. On the next slide, I'd just like to start by saying, who is Avidity and what is our approach to patient-centric drug development? So at Avidity, our mission is really to improve the lives of patients and people affected by diseases with limited therapeutic options, such as Duchenne muscular dystrophy, by advancing a new class of drugs. We are a biotechnology company that's based in San Diego, California. And really, patients and the communities that we serve are at the heart of everything we do. We have a team that brings extensive experience in drug development, and then we combine that with our commitment to involving patients and their families throughout the drug development process. And those two things really serve as the foundation that motivates and drives us to advance treatments for rare muscle diseases, including Duchenne. On the next slide, I'm going to go through a little bit about what we're working on. So we are really working on advancing potential treatments for the DMD community. Avidity's technology has been developed to unlock an innovative way to deliver drugs directly to affected cells like muscle and heart cells. The new technology we're using is being used to develop potential treatments for diseases with currently limited therapeutic options, and we are applying it to develop potential treatments for DMD. As we build our clinical trial program, our goal, as I mentioned, is really to partner with the Duchenne community to ensure that your voice is incorporated and the studies and the things that we are looking into are meaningful, not just to regulators, but also to the people that we are hoping to serve. We are initiating the EXPLORE44 clinical trial with our first program for DMD with a drug potential named AOC 1044, as I mentioned. And Mark's going to get into additional information and share about the preclinical and ongoing research that we've conducted to help support making this available in clinical trials. On the next slide, I'm going to talk a little bit about what I'm talking about. So what is our potential treatment and how is it designed? So our technology is offering a potential new approach to exon skipping. Our potential treatments are called antibody oligonucleotide conjugates. which is a mouthful. So we abbreviate it to shorten it to the first 3 letters of each word, which are AOCs, and they can be designed to target cells such as muscles. We are developing several different AOCs with the goal of treating the genetic cause of muscle diseases, including DMD, as well as other disorders of muscle diseases. The next slide shows the building blocks of the AOCs. So we are combining 2 proven technologies that are well understood actually for what they do and their specific properties in drug development. So the first are antibodies. Many of you are familiar with these. Antibodies are naturally occurring proteins that are made by your body and they help you fight infections. They are commonly used in drug development because they actually can be made to target almost any protein, including those that are different between one cell type and another. And they've been used for a long time as medications and actually are the foundation for over 100 approved drugs, many of which are for chronic conditions. Because of this history, they have a track record and a well understood safety and efficacy profile. And at Avidity, what we're doing is we're using the antibody as a vehicle to deliver our drugs to muscle and heart cells. So we're specifically designing them to get to those cells. Then the oligonucleotide part, or the oligo, which is a short strand of DNA or RNA. And that's the part of the acronym, like I said, that's the oligonucleotide part. And oligos can be powerful drugs because they can be designed to change gene expression or how the body makes a protein by targeting RNA. And there are actually also multiple approved oligo drugs, many of which are disease modifying. At Avidity, we use the oligos in our AOCs tailored specifically to target the root cause of the disease. So on the next slide, I'm going to get a little deeper. The antibody, so as I mentioned, is designed to deliver the oligo into the muscle and heart cells. So the antibody component of the AOC enables that targeted delivery to muscle and heart cells, and in turn improves the power of the oligo piece. The antibody has this unique ability to stick to a specific protein on the muscle and heart cell, delivering more oligo, allowing it to change more gene expression. And so for our AOCs in development for DMD, the antibody binds to a transporter protein called transferrin receptor 1, or TFR1, in the muscle cell to modify gene expression by targeting that RNA. It's an analogy you may have heard before, and one that I have stolen from a colleague, is that you can think of the antibody as a bus that's taking passengers to a specific bus stop where they can get off and go to work where they're supposed to go to work. And in this case, our bus stop is a muscle or a heart cell. So on the next slide, you'll see that we have actually a pipeline of drugs for rare muscle diseases. As I mentioned, we're working in Duchenne, specifically for Exon 44 amenable patients at this moment. We also have a number of other AOC drugs in development, either in the clinic or approaching the clinic, specifically in DM1 and FSHD. And we can move to the next slide. So here at Avidity, we really do believe in patient-centric drug development, and we believe it is at the cross-section of multiple stakeholders. We really do seek input and partnership across the board to develop our new treatments, and believe that it is only through our active and ongoing partnerships across stakeholders, including advocacy organizations like this and the community of families they support, healthcare providers, health authorities like the FDA, coverage and insurance providers, and others, that true patient-centric drug development can be achieved. And these collaborations really help us ensure that people and their families are put first in the decisions that we are making, in advancing our new and meaningful, hopefully meaningful treatment options. So we really do seek input throughout each stage of the drug development process to best meet your needs. On the next slide, I'm just going to go through a few things that are some specifics. So we are really continuously seeking input from the Duchenne community through each stage. We collaborate early and often with the community to ensure that we are doing the right things. We do this through listening, partnering, and connecting with families. We have and do outreach to ensure that we have and understand patient and caregiver perspectives that inform our approach to drug development. And they do really, you inspire us to stay committed to our mission to improve the lives of people impacted by diseases like Duchenne. We design our clinical trials in partnership with patients and their families to ensure we are meeting the needs and their expectations. And we also want to make sure that you feel heard and supported and know that you have an open line of communication to our team. And at the end of the webinar, you'll see an email address where you can reach out to us if you have additional questions. On the next slide, I just wanted to mention, so our commitment to the Duchenne community, again, is rooted in our mission statement, and we believe in involving you in every step of the way. I want to share some important examples of how your voice has already directly impacted our programs. Through partnerships with organizations like PPMD, we are grateful to connect with you in forums like today's webinar to share updates as we move forward, as well as support the important resources and initiatives that they bring to you. We are also invested in gaining a deeper understanding of the journey of living with and caring for someone with Duchenne. Through conversations with you, we've gained important insights into the experiences and also the challenges in your personal journeys to getting a diagnosis, finding the right caregivers, establishing your care team, and how to manage your day-to-day living. We've also brought together focus groups of caregivers and patients from the Duchenne community to inform us, to help us understand what services we need to include in our studies. And finally, we're collaborating with the community leaders in an ongoing way with the goal of really addressing any gaps in support services and resources that are available, and hopefully bring those to you. So thank you to PPMD and the community you serve. We are grateful for your partnership and hope that we are working towards our shared goal of bringing new therapies to the Duchenne community. With that, I'm going to hand over to Mark, who's going to get to the meat of the things that I think everybody wants to hear about, the AOC 1044 and EXPLORE44 clinical trial.
All right, thanks, Kelly. Hello, everyone. I also want to say thanks to PPMD for the opportunity to speak with you all. My name is Mark Stahl, and I'm a neurologist and a scientist working on developing new medicines here at Avidity Biosciences. Go to the next slide. As Kelly mentioned, Avidity is developing multiple exon-skipping AOC drugs, and we'll talk a bit more about exactly what that means momentarily. Our first program for the treatment of Duchenne is called AOC 1044, and it's designed to treat individuals whose mutations are amenable to exon 44 skipping treatments. We are now advancing AOC 1044 into a phase 1/2 clinical trial, and I'll talk about that momentarily. We expect to learn from the development of AOC 1044 and then apply that knowledge to our other programs that are currently in the research stage, as you can see here. On the next slide, I'll go into the science of why these exon skipping treatments really do depend on an individual's DMD mutation. So this is exon skipping. You know, what's the work that the oligonucleotide actually does in the muscle? It's that. So many of you are probably familiar with this idea because there are already drugs that do this, but we can review. What exactly is exon skipping? So this sentence is a metaphor for exon skipping. You might remember from maybe high school or college that the genetic code is a triplet code. That is, it's made of a series of 3 chemical letters that tell a cell what part of a protein, what individual amino acid, to add to the growing chain that eventually makes a full-length protein, like dystrophin in this case. When part of a gene is deleted, that deletion can cut off whole words, like in the first example. This is called an in-frame mutation because the remaining words are unchanged. This leads to a sentence that's missing words but still makes sense, or a gene message that makes a shorter but still usable dystrophin protein. This kind of deletion happens in dystrophin naturally and is what we see in Becker muscular dystrophy. The second sentence is what happens when a deletion cuts off part of a word. Because the genetic code always works on 3 letters, parts of words get like smushed together and you end up with garbled nonsense. When this happens to a gene, the cell will literally delete the message that the gene makes at the end. That's why people with Duchenne make almost no usable dystrophin protein. The last sentence in purple is what happens with exon skipping. An oligo can force the cell to skip over reading part of the gene and give you a message that is shorter, so not perfect, but that is still readable. So in terms of Duchenne, you're now able to make this shorter dystrophin protein where before you had almost none. As you might also be able to tell, skipping a specific part of the sentence only gives you a readable outcome for very specific deletions. And that's why exon skipping depends on a person's specific mutation. So exon 51, exon 44, and so on. So that's the science lesson, and I hope that makes sense to everyone. And that's what exon skipping really does. So if we can go to the next slide, we'll talk a little bit about how what we are doing is different from the exon skipping that you might already be familiar with. As many of you know, exon skipping is the technique that's used by 4 currently approved drugs. But those drugs are not targeted to muscle like AOCs are with our antibody. So without the targeted delivery that the antibody provides, oligos have inefficient uptake by muscle and heart cells, and so they don't make all that much dystrophin, and they require frequent dosing. This is what we aim to improve upon with our technique. So we'll go to the next slide. Kelly very nicely introduced this already, but let's review the idea of what our drug can do for Duchenne, and then we'll talk about specifics. Remember that AOCs combine antibodies that target muscle and heart cells with exon skipping oligos. So in this diagram, you can see the AOC on the left. The antibody part of the AOC finds the specific protein on muscle and heart cells that Kelly mentioned, the TFR1, the transferrin receptor 1, to deliver the oligo into those specific cells. Once delivered, the oligo enters the cell to modify gene expression by targeting the RNA message that eventually makes protein. So by combining the oligo and the antibody, we're able to more efficiently deliver the oligo directly to muscle and heart cells. More oligo means more skipping, which means more dystrophin. Let me show you some data from our labs to get started. Here's the next on the next slide. So let me show you a little bit about the expected effects our AOCs have based on preclinical or laboratory experiments in the next few slides. Here in this experiment, we tested just the exon skipping ability of our oligo that skips over exon 44 in muscle cells from DMD patients, showing that it works well when cells are exposed to it directly. This is a nice demonstration of the dose effect on the amount of exon skipping. So as I said, more oligo in the cells means more exon skipping, and this experiment shows that. You can go to the next slide. Here, we're comparing what happens when that highly effective oligo is given alone, so as an unconjugated oligo, versus when it's connected to an antibody in a mouse model of Duchenne. Here you can see just how much exon skipping we get with the oligo alone in the blue on this graph, and that's around 1%. And then you can see here, when in the green, when we get that exact same amount of the exact same oligo, except connected to an antibody to make an AOC. Having the antibody portion of the drug increases exon skipping by about 50 times over having an oligo alone. That's a result we found very exciting. You can go to the next slide. Here we looked at what that skipping means for the very important task of actually making dystrophin protein. So you can see here, we again compared single doses of the same amount of oligo with the antibody in green and without the antibody in blue. We see an increase from about 1 to 3% or so of normal levels of dystrophin, up to about 20 or 30% in skeletal muscle and a bit less than that in heart. Now remember, these are single doses, and we may expect to see accumulation of dystrophin when we do multiple doses in other animal models, and then of course in people. And we go to the next slide. In the last set of these laboratory experiments I'll show you today, you can visually see both that the amount and the distribution of dystrophin made with the AOC treatment is very close to that is what's seen in the normal mouse. The distribution of dystrophin protein evenly throughout the muscle may also be very important for muscle function, and is really quite nice with the targeted nature of the AOC, instead of relying on the naked oligonucleotide to just passively diffuse from the muscle into the tissue, where areas near blood vessels may get more, areas far away from them may get less. We get a nice, smooth production of dystrophin throughout the muscle fiber, which means that it will work as a unit much better than if there were patches of areas with high dystrophin and areas of low dystrophin. Okay, that's enough of the science lesson for now. Let's talk about the study. You can go to the next slide. As you can see from the title slide, this is what's called a phase 1 and 2 study. of our drug, AOC 1044, in two parts, one in healthy volunteers and one in people who have the DMD mutations that are amenable to exon 44 skipping. If we go to the next slide, we'll talk a little bit about the study in more detail. Now, the EXPLORE44 study was designed in partnership, as Kelly mentioned, with patients, caregivers, advocates, and key physicians in the field to ensure we are best meeting the needs of the Duchenne community. This study starts with Part A, which will evaluate single ascending doses, so increasing amounts, of AOC 1044 compared to placebo in about 40 healthy volunteers. Part B is then planned to enroll 24 participants with DMD between the ages of 7 and 27 years, regardless of ambulatory status. So we have both ambulatory and non-ambulatory participants invited. Part B will evaluate multiple doses of AOC 1044 compared to placebo, administered via an IV infusion that we give every 6 weeks. The primary goal of EXPLORE44 is to evaluate the safety and tolerability of AOC 1044, but will also include tests of dystrophin production as measured by muscle biopsy, and then muscle function, patient-reported outcomes like how people are functioning in your daily activities and abilities and so forth, and then quality of life measurements, all in the participants who have Duchenne. If we go to the next slide, we have a diagram that I think explains this reasonably well. Great. This slide shows an overview of the study design. So in the darker bars on the left, you can see Part A of EXPLORE44. That's in the healthy volunteers. This is where our 40 volunteers receive single doses that are of AOC 1044 or placebo via an IV infusion. And we increase the dose to test the safety and tolerability of higher and higher doses as we progress through the study. And then in Part B, you can see in the teal bars on the right, 24 participants with Duchenne will receive 3 doses of either AOC 1044 or a placebo via an IV infusion, as I said, dosed every 6 weeks. In EXPLORE44, 3/4 of the participants with DMD receive AOC 1044, and 1/4 of the participants will receive placebo. The plan then is to offer all of the Part B participants the opportunity to continue in a follow-up extension trial. So you can see in the orange, the extension study that would follow on after EXPLORE44, where everyone, including those who start on placebo, would receive AOC 1044 every 6 weeks for a longer period of time. The 2-part design of EXPLORE44 enables efficient safety and tolerability testing with all the associated blood draws and EKGs and so on in healthy volunteers, while minimizing the burden on participants who have DMD by not giving them doses that we anticipate to be lower than what would help produce dystrophin in a patient. Overall, we think this is the fastest, safest, and really most considerate way we can bring this therapy to the patients who need it. We go to the next slide. I just want to point out that there are multiple features of the study designed to make the patient experience as positive as it can be, and most importantly, to ensure that families can participate. So you can see here, 3 of the most important factors there include some of the visits that are designed to be conducted either in the clinic or via home health. So having a nurse come to the home to maybe take blood or do EKGs or things that don't need to be done in the clinic. We also have engaged with a travel concierge who can help set up all of the logistics of airfare, lodging, transportation, and things like that. And then, of course, the ability to reimburse the participants for reasonable expenses. And so all of those things are designed to make the patient experience a positive one in our study. We go to the next slide. So just a few key takeaways or points in summary. Number one, we are extremely excited by the potential that we see in Avidity's AOCs to treat the underlying cause of Duchenne in muscle and in heart. AOC treatment has demonstrated robust exon skipping, restoration of dystrophin protein, and improvement in muscle function in mouse models of DMD. And with our IND application now cleared, we have started our first clinical trial in Duchenne called EXPLORE44 with patients with dystrophin mutations that are amenable to exon 44 skipping. We are planning for IV dosing every 6 weeks in our study. And we plan that what we learn from our lead program in Exon 44 amenable patients will then inform and streamline our programs for the other exon skipping drugs that we're working on. Avidity is partnering with the Duchenne patient advocacy community to get more information and resources to you as soon as we have them available. And of course, with our next slide, one of my favorites, here are some of the Avidity team members who are working hard every day and that are looking forward to connecting with and hearing from you. So please feel free to reach out to us with questions at the email address at the bottom of the screen here, patients@aviditybio.com. Thank you so much for your attention.
Thanks, Mark. Thanks, Kelly. Just to everyone who's watching, so we are going to start to the open Q&A. So if you have questions, please submit them to the chat below. I've seen a number of them kind of start to come in, and so we'll jump into that. The first one I'm going to ask, I'm also going to make a little plug for PPMD and our website. So there was a question about a specific deletion and whether their child might be amenable to AOC 1044. PPMD does have a Duchenne exon skipping tool on our website. It's under the About Duchenne page, and I'll make sure that we put a link in the chat and with the whenever the video is posted on the website, where you can put in your child's deletion or your deletion and see if it could align for what type of exon skipping you might be amenable for. And Mark, I don't know if you know off the top of your head if exon 45, 54 might be likely.
I do actually know off the top of my head that yes, that is considered an exon 44 skip amenable mutation. For the specifics of whether you or your child would be appropriate for a specific trial, of course that's a discussion to have with your physician and perhaps to review your genetic testing. But in general, yes, 45 to 54 is considered exon 44 skip amenable.
Yes, and we do always recommend have those conversations with your care team. They know a lot. They know a lot about these trials. They're a great resource. And you can also contact the genetic counselors at PPMD to talk through some of those pieces as well. There were a few questions around kind of the age within the trials. There was a question essentially around the age limit of 27 and what the rationale was for kind of capping at that age.
Yeah, that's a good question. The age range of a trial really is designed to use the initial information that we have from studies that like I've shown you in cells and in animal models and things like that, and translate them the best that we can into the population that we think will be the most informative really for the very first initial studies in people. And then of course, as I'm sure most of you are familiar, in order to really translate early findings into the larger community, the expectation is to go to generally larger and larger studies that demonstrate clear improvement and benefit on kind of function and strength and things of that nature. And so with this initial study, we are really looking to gather the initial information about exactly who will benefit and exactly how they will benefit from our drug. And so we really are looking at a relatively restricted patient population to start, so that we can use that information to see exactly where we can go next. The choice of the age range has a lot to do with how we expect the drug to distribute to the muscle and what we'll be able to see in muscle biopsies, and then what kind of information we can gather about the functional tests that we'll be doing going forward. And so in Duchenne, there are certain characteristics that we'll expect to see of patients that are of representative ranges, and that's kind of where we need to concentrate to get the information that we need to inform the next study that goes forward. So there there's a little bit of kind of projecting based on what our, kind of early studies and modeling tell us will probably give us the most information going forward.
Great, thank you. And then a question around, has the healthy volunteer study started?
It has indeed.
Okay. And then, of course, with some immediate follow-ups, when is, if you can share, would enrollment start for the Duchenne patients, and if you have kind of sites in mind for where that might be.
So I can actually, if you want to move back a little bit, maybe like 3 slides. Yeah. Sorry, forward one more to the diagram. Yeah. So as you can see here, the way that the study is designed is to evaluate, safety and tolerability of the drug in the healthy volunteers. And so the opening of the initial patient portion of the study does depend on getting through some of the initial healthy volunteer groups. And so, what the upshot of that is that I can't tell you an exact date when we're going to be able to open the patient portion of the study, because it's dependent on how the recruitment and progress through Part A goes. I can tell you that it's started, but unfortunately, I can't tell you exactly when to expect the next portion to begin.
That's fair.
Right, there was another question. Sorry. Was it about sites?
Sites, yes.
We are actively working to make our site selections. That's a process actually that is very much ongoing at the moment. And in fact, I will because we have the magic of virtual visits, I'll actually be conducting one of the site visits later on today. But yes, we are looking very carefully and working very hard on making our site selection for Part B right now.
That's great. And as soon as Avidity does make that public, we will be sure to share as well on PPMD's website, so that way you can start to look into that and again have those conversations with your care team about potential for joining. We did have a number of participants from kind of international locations asking about potential to join, or I guess, Avidity's thoughts in terms of maybe following this, what your plans are for expansion outside of the US, since I think as of right now, it sounds like mostly looking at US sites for this trial.
Yeah, that's right. Like I say, we don't have all of the sites, entirely selected yet. So this isn't really quite final, but do keep in mind that this is a small initial study that will be followed up, with a larger study that we expect to be, more broadly, outside just the United States. But this initial study, because of the size, is mostly expected to be in the United States.
Okay, great. With regards to the extension study, is that when patients finally do move into that, are all of the patients going to be moved on to a similar dose, or are they going to be kept on the same dose that if they were previously on either the 5, the 10, or the 20 for that time?
That's a great question. Yeah, that's a great question, and that has not been entirely finalized, but I will say that the initial plan will be to decide on a single dose that we would move all of the patients to that is well tolerated and that we expect to be effective, and that would be the dose. But it is also possible that we'll have information that from the initial study that would suggest that we should evaluate, say, more than one dose. So that could also happen. But the current plan would be to identify a single ideal dose that would cover everyone.
And would that largely be based on some of the biopsy data that you collect?
It would be based on the totality of all of the data that we have. So safety, tolerability, how patients are doing on some of the, early functional stuff, just everything that we're able to gather.
Okay. And with your protocol, have you landed on the number of biopsies and whether those are needle or open?
These would be open biopsies.
Okay.
Yeah.
Um, so we did another question. I think maybe someone, if they had joined late, but you had mentioned that in this study for the 7 to 27, ambulatory status is not a factor. They could be ambulatory or non-ambulatory.
That is correct.
And then I have some of the other ones that come in a lot. Just questions around things like steroids. Is that going to be a requirement for a study like this, kind of steady state, or can patients?
Yeah. Good question, as always. In general, we would only require that, and this will be, sort of in the specifics of the inclusion criteria that you would go into. But overall, we would really just require that the steroid dose be consistent or not expected to change over the course of the trial.
Great.
If you start off not on steroids, we would hope, our expectation would be that there's not a plan to begin them. Mm-hmm. And if you come in on steroids, we would hope that we would be able to maintain that at a consistent level.
Yeah. And then one thing you mentioned, and I do think is something to point out again, is that your dose schedule for this is 6 weeks. So I think most people are used to, when with the approved products and kind of the just ASOs on their own, a weekly infusion, or some of the other conjugates that we've started to see be developed is a monthly infusion, and you're at every 6 weeks. And so you just talk us through how you arrived there. Was that from some of the preclinical animal model data you did? Was that looking at patient burden and trying to minimize some of that aspect?
Yeah, exactly. That's a great question. So, one of the things that really we heard very much in discussing with patients and really getting, patient input on the study design, which of course, as Kelly and I both, I think, mentioned, is a very important thing for us here at Avidity to get patient input on and family input on our studies, was that it is so burdensome, right, to have very frequent dosing and require, that all of these are IV infusions, right? And so that our plan was to go in with the the longest interval between doses that we reasonably could, while still maintaining as much efficacy as possible. And so that was an intentional design choice to maximize that interval. Of course, that is based heavily on what we could see with dystrophin change and with the distribution of the drug in our preclinical studies. But the ultimate choice on dose interval really was to take what seemed feasible from those preclinical experiments and translate that to the longest possible interval with the least possible burden on the families and patients themselves. So yes, that was an intentional choice, and I will say that part of that, the ability to do something like that and to have a longer dose interval is down to the technology, wherein we use the monoclonal antibody, which has a little bit longer half-life or time that it sticks around in the circulation, kind of enables something like that. So we are really excited by the fact that we're able to offer a longer dose interval than might have been possible with other ways of accomplishing the dystrophin increases that we're seeing.
And then we actually had a question about on the slide that has showing some of the dystrophin expression in mice. Someone noted, the expression of dystrophin in the heart was lower than what you see in some of the other muscles. And is that something that you think will increase the longer you're on the drug? And I think you mentioned this is a single dose here.
That's right. These are all single doses. And so one of the, this actually does dovetail pretty nicely with the previous question. One thing that we did design the dose interval to do was to have the next dose in a time frame that would allow for the accumulation of dystrophin, right? So dystrophin is a reasonably long-lived protein, and even dystrophin that's internally truncated or is shorter than the normal dystrophin still sticks around for a pretty long time. You know, although there's not an exact single number, because all of the dystrophin you know, that comes from different mutations will be a little bit different. You know, it can be around for, you know, up to maybe 3 months or so, maybe a little bit longer than that even. And so so with the 6-week dosing interval, we absolutely expect that there will be you know, an accumulation of dystrophin, and that ultimately we'll only see, you know, the final you know, sort of highest levels that will be achievable after multiple doses. And so that's why our biopsies will be scheduled sort of out after multiple dose intervals. But yes, we will expect that all of these you know, amounts of dystrophin should increase over time. I will, you know, make a little bit of a caveat though. You know, this is in mice. These are not necessarily, you know, the exact translated numbers that we, you know, that we'll see in people. That you know, that's not necessarily a one-to-one. You know, oh, we see, you know, 30% dystrophin restoration with a single dose of this drug in mice. I don't want to give anyone the impression that that's, you know, we expect to see a single dose of our AOC 1044 to result in 30% dystrophin restoration in humans necessarily. We don't know what that will actually end up being until we do some biopsies in people. But I do want to just caveat that this, again, this is a mouse experiment that gives us sort of an idea, but not to, you know, not to directly translate that number literally.
Right. I mean, this is why we have to go and do the clinical trial piece and actually get the data and find out. And I actually think this is since I have you here and you have some experience with this, a good drug development kind of education moment, because there was another question around this and looking at mice. And I think looking through the two different types of experiments you did is also helpful for understanding that process. Because the first one you did was kind of the more proof of concept with just muscle cells from a patient, not in an environment, but just kind of alone to see if you can get that skipping.
Exactly.
And then so that's good proof of concept, but again, that's not in kind of the environment of a systemic delivery into a person.
That's right. These are just cells in a dish, so we have to make that clear. Yeah.
But it does give that proof of concept that this is something that can do the exon skipping.
That's right.
And then in your mouse ones data, I think it's also just, it'd be good to point out that you're not skipping exon 44 in those models, correct? Because this is not a small molecule. You're not just giving a steroid. It's not the same steroid to the mouse and the human. These are genetic therapies, so you do have to tailor it. So how different is what you're looking at in the mouse versus what is being delivered in the clinical trials?
Thank you for this. This is a great question. So if we actually go back just one slide, just to really make this clear. Yeah. So this experiment was to test the ability of the oligonucleotide, the actual oligonucleotide that we will use in people, to test the ability of that sequence, right, that oligonucleotide sequence, to cause exon skipping in human cells, right, in the cells that are donated from, you know, and come from people who have Duchenne. And so this proves that, you know, when we connect our antibody to this oligo, that assuming the oligo gets into cells, that it will cause the exon skipping that we want, right? So that's sort of one piece, right? And that's a separate piece from the next slide, which is how much does it matter to have an antibody on an oligo versus not have an antibody on an oligo? But the question that we're answering here is just generically, right? So this is a mouse, right, using a mouse antibody, right, to an exon that's actually exon 23 in the mouse dystrophin, right? So this is not, you know, this is very much not, you know, our drug, right, at all. But it does evaluate a very important concept or answer a very important question, which is we kind of have an idea of what happens when we give, you know, these naked oligonucleotides systemically in animals and in people, right? Because we now have 4 drugs that are approved that do that, right? So what is the improvement, or is there any improvement between having a naked oligonucleotide, which we kind of know a little bit about and how they work, and versus connecting it to an antibody and making an AOC, right? Now that doesn't mean that this is our AOC, it's not, but what is the effect of having an antibody attached to it? And we can see that, when we, all things being equal, right, a naked oligo gives X amount of exon skipping, and then an antibody connected oligo, so an AOC type drug, gives 50 times better. So whatever you were getting before, it's this much better, right? So that's the kind of takeaway from an experiment like this. But it is absolutely not the same as saying, oh, our drug is this, right? That's not quite the same thing. So I do want to really emphasize that these are experiments in mice, and they are very, very encouraging. But of course, they're still experiments in mice, and we really do need to see what happens in people.
Yeah, but this builds that evidence and confidence, hopefully, as you go into that.
Exactly.
And just the very last thing I wanted to touch on, because I don't think we don't see this a lot in the Duchenne space, in our rare disease space, of having some of these healthy volunteer studies, and especially one in this sense where it is kind of in the same phase as you're transitioning through. So can you just explain just the rationale for including healthy volunteers rather than just going straight to the boys and the young men in this case?
Absolutely. So actually, it turns out that most drugs, even the naked oligonucleotides, had been tested in healthy people, at least to some extent, before coming to patients, and that's actually a pretty common process, right? When we have a new drug, right, something that's not been done before, it is actually extremely common to test it out in healthy people first. And there's two reasons why we chose to do it this way. If you want to actually call up the slide for the in our study. So the first is that, unlike sort of the naked oligonucleotides, this really is a new type of drug, right? I mean, Kelly talked a little bit about how we know a lot about how antibodies work, and we do, and that is a huge advantage in being able to predict how our medication might behave and things like that. And oligonucleotides, of course, as we know, are already quite a few drugs, and having that as part of our drug is a huge advantage, again, partly for understanding it. But it has never been there has never been a drug that put them together, right? So this is the very first one. So this is something that we really did need to test and make sure that there was nothing unexpected or anything like that. And so doing some studies of that in healthy volunteers is important, just to kind of be certain of that what we expect from experiments in animals and cells, and from the components of our drug, that those really do translate when we put them together, right? So that we don't run into anything unexpected. But actually, I would say the perhaps more important thing is that you can see the doses, right, that are listed here. It starts at 1.5 mg per kg, goes to 3, 5, 10, 20. Well, our modeling suggested that there was not a lot of benefit, in terms of exon skipping or dystrophin, that would come from the doses that are low, right? I mean, there may be some, but our goal was to really only ask people with Duchenne to participate in a study that they would have a good chance of benefiting from directly, right? Not just like, it's good to do for the community and all that, which of course is important, but we want the participants to have at least a reasonably good chance of having direct benefit from the drug. And so we wanted to make sure that the drug was well tolerated and safe, but at these low doses, there was really not much benefit to the individual DMD participants. So we wanted to test those low doses in healthy volunteers, so that we could really concentrate the high doses that we thought would be effective in people with Duchenne, and then this also has some other benefits. For example, we can do pretty intensive testing, like do a lot of blood draws and a lot of EKGs and all that kind of stuff that really takes a lot of time and effort in healthy volunteers, and gain a lot of information about our drug that way, and not have to burden the families of folks with DMD with doing all that testing and taking all that blood and all of those kinds of things that are kind of a drag, quite honestly, and not have them burdened with that if we could get that information elsewhere. And so we really feel like there's a tremendous advantage to kind of this hybrid approach where we get a lot of the necessary testing almost kind of out of the way, before we ask people who actually have Duchenne to participate in the study, but yet we still get it to them as fast as possible because it's an integrated study. So once we get into we only have to go partway through the healthy volunteer portion to get into patients. So that you could have said, which many other ways of doing drug development would have done, do the study that is listed as Part A as a separate study, finish all that, analyze that data, talk to FDA, and then start Part B. But that would actually probably delay things by a while, months to years maybe, depending on how things went. So we wanted to kind of combine those approaches to relieve burden, get a beneficial drug into patients, and do it as quickly as we could, all in one study. So it makes for some, it's complicated, admittedly, but it probably, I think, is the best way to kind of get the fastest, safest, and really most, I guess, patient-centric way of doing it, at least that we could design for our drug.
No, we always appreciate innovative design and ways to reduce burden on the patients. And then I think one final question was just to clarify with the extension study, will patients immediately roll over into the extension study after their observation, or is there going to be a wait and kind of a drug holiday before the extension study would start?
Excellent question. Thank you for asking that. First, I will emphasize again that the extension study, is in the future and has not been finalized yet. So this is, again, based on what we currently understand of what the, both the regulatory requirements are and what the data should, be from the initial part of the study. But the plan would be actually to immediately roll over and probably continue on the dose that was in the assigned cohort. So in other words, if you were on 5, when you rolled over to the extension, you would probably stay on 5 for a period of time until we had enough data to decide what that dose for the extension would be. So that could be, for the very earliest patients, that could be some while, because some months, because we'll want to get data from the second cohort and the third to make that decision on what that chosen dose is going to be, or potentially more than one, but probably just one dose going forward. So that could be a little while that you would just stay on your initial assigned dose. But I want to emphasize that none of these, again, as I said with the last answer, we expect all of these doses to at least potentially have direct benefit. So hopefully that's even needing to stay on that dose for some period of a few months while the rest of the study continues, will still be providing benefit to you.
Well, great.
Hope that makes sense.
Yeah, no, I think that was very clear. And as information comes out, as study sites get up and running, and you're able to start enrolling, and you're able to share that information, we of course will share that with the community as well. Mark and Kelly, thank you both for joining us today. I'm glad we as well got to do a little drug development education piece in there. I think that's great. We don't get to talk about that a lot with the community, and I think it's really helpful for families as they try to understand some of this data that they see and how that actually is going to be, kind of impact to expect and things like that. So that was great, and I really appreciate that. For everyone who joined us today, thank you for sharing your questions with us. This is recorded. We will be posting it to PPMD's website sometime within the next week. So if you have any friends or family that this would be really relevant to, please feel free to let them know and share them. But again, thank you, Mark and Kelly, for joining us today.
Thank you. It was a pleasure.
Thank you.